Hello, everyone, and welcome to today's panel presentation titled Achieving Efficient and Sensitive Rare Cell Capture for Culture and Downstream Molecular Analyses. This webinar is a part of the 11th annual event in the Genetics Week virtual event series. I am Sydney McNeil of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by Bio-Rad. For more information about Bio-Rad, go to www.biorad.com. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the left of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many questions as we have time for at the end of the presentation. If you have any trouble seeing or hearing the presentation, click on the Help Desk button located at the top of your screen within the navigation bar or from the lobby within the event. Finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on the continuing education credits link located in the abstract tab from the menu at the left of your screen and follow the process to obtain your credits. Now let's get started. I now present today's speakers, Mina Zeinali, Single Cell Field Application Specialist at Bio-Rad Laboratories, and Cissy Jiang, Senior Global Product Manager of NGS Products at Bio-Rad Laboratories. For a complete biography on our speakers, please visit the Presenter tab from the menu on the left of your screen. Mina and Cissy, you may now begin your presentation.
Thanks, Sydney, for the introduction. Today, we will cover a short introduction of the concept of liquid biopsy with circulating tumor cells, also known as CTCs. We will go over the existing CTC capture technologies and the challenges involved. Then introduce you to our new CTC capture platform, the Genesis cell isolation system, and the Celselect slide technology. And in the end, we will cover some downstream molecular applications. Liquid biopsy refers to the identification of tumor-derived materials in body fluid, including in blood circulation. Liquid biopsy offers many advantages for cancer detection and monitoring, which include its non-invasive, low cost, potential for genomic testing, ability to monitor tumor evolution throughout treatment, and it includes heterogeneous cell types. Blood contains many different biomarkers in the plasma and cellular fractions, including CTCs, extracellular vesicles or EVs, protein and circulating tumor DNA or ctDNA, and other markers. It's worth considering each of these biomarkers separately as they provide different and potentially complementary information. However, in this talk, we will focus on single CTCs and CTC clusters. Cancer metastases represent the spread of tumor from a primary site to distant location. During this process, cells intravasate from the primary tumor, enter the blood circulation, and then extravasate to form secondary tumors, as you can see in this figure. CTCs are viable circulating biomarkers that function as seeds of metastasis. They consist of heterogeneous cell types and represent heterogeneity within the tumor. Unlike conventional biopsy, they may be sampled through simple blood draws. Analysis of CTCs can be complementary to the ctDNA workflow since they are ideal markers for both cellular and molecular assays to study cancer progression and metastasis. CTCs are challenging to capture, but they remain highly desirable as biomarkers since they are useful for screening for early detection and tumor recurrence, monitoring for response to therapy and patient's prognosis, as a real-time biopsy since it's minimally invasive and is repeatable, and personalizing medicine by identification of targets and ex vivo therapeutic targets. However, they are extremely rare and only a few CTCs per billions of blood cells, thus making their isolation and characterization a tremendous technical challenge. So as you can see here, it can be like finding a needle in a haystack. So let's solve the problem as scientists, but unfortunately, we cannot burn this haystack. There are two main approaches that have been used for CTC isolation: label-dependent and physical capture. In label-dependent captures, they're using affinity reagents to target as surface proteins on cells. This was previously widely used for its highly sensitive and high purity isolation, but this method has low throughput and can also produce biased cell selection. For example, the first FDA-cleared CTC technology, cell search, they only use anti-AbCam um, markers expression that has poor applicability to many blood-based cancers, including melanoma, amyloid, and etc. However, physical capture method exploit differences in properties of cells, such as size, deformability, and density. These methods include, for example, filtration, Fickel gradient centrifugation, electrical field separation, and many others. These methods are often capable of higher throughput, and the user can obtain a heterogeneous CTC population. However, some small cells, CTCs can be actually lost during this process. The main limitation of existing methods are low sensitivity, long processing time, low capture efficiency, low throughput, and also incomplete solutions that don't cover all desired applications. The Genesis cell isolation system has been developed to address these key challenges of rare cell capture by ensuring high capture efficiency, increased sensitivity, and increased sample throughput. The system is compatible with multiple applications and is also flexible with customizable end user protocols. The system works with applying microfluidic size-based separation using Celselect slide technology. I'm going to walk you through this graph. As you can see here on the left side, you can see the Genesis system, which is a benchmark system, and the blood sample can pass through the inlet area, going through the Celselect slide chip, and then the unwanted cells can go to the waste jar. And as you can see here on the right, the cells of interest, the blue one, as you can see here, can be captured inside the microchambers, while the other unwanted cells, like RBCs and white blood cell, can pass through the microchambers. Technology enables the isolation of CTCs from liquid biopsy samples, such as blood, as I mentioned that, and there are two sample lanes, as you can see, enabling the processing of two samples simultaneously. Celselect slide is designed with 56,400 microchambers that capture CTCs based on differences in cell size and deformability, while RBCs and white blood cells, shown here as red and white, filtered out throughout the microchambers, and while the CTCs, the blue one, can be captured throughout the chips. The platform enables two different automated applications on the same system. Enumeration is for counting the number of captured CTCs using fluorescent staining protocol, as you can see here on the left, and enrichment for the retrieval of viable CTCs for downstream analysis, as it shows on the right. For today, we will share a proposed workflow to evaluate rare cell capture using the Genesis platform, which can provide prepared samples for either cellular analysis or molecular analysis, or both, depending on the experimental design. In the interest of time, we will focus on molecular analysis, including gene expression profiles and gene mutation analysis, which can be done using other Bio-Rad products such as CFX Opus for real-time qPCR, the Sequoia kit for RNA library prep, and the QX600 for the digital PCR. The Genesis platform includes the protocol builder and rare cell analysis software, which are both available on the website. The protocol builder is a flexible tool that can be used to customize system parameters such as, for example, reagent dispensing, incubation time, pump pressure, temperature, and etc. This makes it easy to develop and run custom protocol on the Genesis system. And the rare cell analysis software has been recently released to identify cells on Celselect slides based on a staining panel and morphology. It is compatible with images taken from any automated microscope and analyzes images with up to 6 fluorescent channels. Now we would like to talk about some of the performance verification results our Bio-Rad team has generated. Here are some fluorescent microscope images of single CTCs and CTC clusters captured on a Celselect slide. As you can see here, cells positive for DAPI, which is in blue, pancytokeratin, which is in green, and they are negative for CD45, which is red, are identified as CTCs by the software. So as you can see here in this graph, the top panel shows a single CTC and a white blood cell in green and red, and the bottom panel has a smaller cluster of 2 CTCs that they have been captured inside the chamber. In this set of experiment, breast cancer cell line MCF7 was spiked into healthy donor blood and processed using Genesis. We have validated a high capture efficiency of greater than 80% using a range of blood volume from 4 ml to 200 microliter. And on the right, we can see that the capture efficiency was highly linear for samples containing a low concentration of 10 cells per ml up to 1,000 cells per ml. Using the same spiking experimental method, we evaluated the percentage of background blood cell depletion from enriched cells using Genesis. In figure A, you can see the blood cell removal rate was 99.9% from whole blood. In B, when we were using only buffy coat separation, the white blood cell removal rate was 99.8%. Figure C shows that Genesis capture efficiency was fairly consistent using various type of blood collection tubes, including EDTA, Streck tube, cell save, cell rescue, and DNA packs. As I mentioned earlier, Genesis system isolates CTCs based on their physical properties. So is basically size separation technology, right? So we have shown that these cell line experiments that most cancer types, when they are in the range of 8 to 30, can be isolated with high efficiency, including breast, melanoma, prostate, pancreas, glioma, and lung. And in this table that the list of cell lines has been listed if you're interested in to know which kind of cell line that has been used. Even leukemia cells, which is not a solid tumor, and which are also very small, can be captured using the platform. Meanwhile, we are trying to increase this number, as you can see here, compared to the rest of capture efficiency rate across the other cancer type. This is a little bit lower, but we are working to increase this number by employing other techniques, so stay tuned. This figure shows that the majority of the captured cells can be retrieved from Celselect slide using the enriched protocol and made available for downstream application. Now, finally, I'm going to present several of our research publications that not only highlight the diverse application of the Genesis system and Celselect slide technology, but also demonstrate the previously mentioned advantages of the Genesis system that overcomes the current limitation of CTC enrichment and enumeration technology. This study highlights the versatility of the Genesis system. This group of people have used different breast cancer cell lines, including epithelial cell type, such as MCF7 and SKBR3, and mesenchymal cell type, for example, MDA, MB231, as well as custom panel of biomarkers, including pancytokeratin, HER2, ER, PI3K, A-caterin, E-caterin, and vimentin. And as you can see here, due to the fact that Genesis is a size-based separation platform, we won't miss different CTC population. We're capturing both epithelial as well as mesenchymal CTCs. In this publication, the capture of CTC using the Genesis Celselect system and cell search was evaluated by this group of researchers. The authors showed higher number of CTCs in more clinical samples using Celselect compared to cell search. They have confirmed that these cells correctly identified using PSA, and the paper suggested that Celselect CTC technology appears to perform superior compared to cell search. This study highlights the capture efficiency and reproducibility of the Genesis system by assessing its sensitivity with a spiking study using non-small cell lung cancer, NCI 441. Researchers found that even when they are come to even 5 CTCs, they are able to efficiently and reproducibly capture these cells. This was confirmed using a custom antibody panel that include cytokeratin, PD-L1, as well as CD45. Furthermore, this group analyzed 10 non-small cell lung cancer patient tissue sample with paired blood samples for CTC enumeration, and they found a strong correlation between CTC numbers and the frequency of immune checkpoint marker expressing lymphocyte, as well as a strong correlation between CTC numbers and the frequency of PD-L1 expressing cancer cells and cancer cell DNA content. In the following study, researchers investigated epithelial mesenchymal transition, or EMT status, from metastatic breast cancer patients who had received eribulin-based treatment, which reportedly suppresses EMT as a means of tumor suppression. They were using a custom antibody panel to count both mesenchymal CTCs as well as epithelial CTCs. They have shown that not only low CTC counts correlated with survival, but also counting both epithelial CTCs as well as mesenchymal CTCs have better prognostic indications. In summary, the Genesis Celselect technology offers a unique solution for highly efficient and specific CTC capture. By using size-based selection, heterogeneous CTCs and CTC clusters population can be isolated from whole blood. Genesis offers two automated applications, which is a unique features in the market. It can enrich CTCs and retrieve viable CTCs for downstream analysis, or stain cells directly on chip for enumeration. Sample processing is rapid with minimal sample preparation required. And at the end, Genesis is also a flexible platform that is highly customizable for user-defined protocol using the protocol builder software. In the next two slides, I would like to talk about the evaluation of downstream molecular analysis using enriched CTCs from Genesis. This section has two parts. Part one is focused on mutation deletion by droplet digital PCR, and my colleague Cissy will continue with the gene expression by quantitative PCR and NGS sequencing in part two. In addition to conventional CTC enumeration studies, Genesis is compatible with downstream molecular analysis, including ddPCR. In this specific study, we spoke in two different lung cancer cells into a blood sample. A549, which is a wild type with no EGFR mutation, and H1975, which has 2-point mutation, L859R, and T790M. CTCs enriched by Genesis have been processed using Bio-Rad QX200 droplet digital system, and as it shows on the right graph, the cells with points mutation are detectable, while no mutation have been detected in the wild type cell line in the left side graph. Our collaborators at Biodesix used the Genesis system to isolate CTCs from 6 non-small cell lung cancer patients. This was a combined study of both CTC as well as ctDNA workflow, as cells were isolated from a Streck tube, and the sample after the sample was collected for ctDNA analysis. The enriched cells were processed using QX600 system for ddPCR, showing that expression of immunotropy marker PD-L1 was detectable in both early and late stage patients. Now my colleague Cissy will continue our talk on the evaluation of downstream molecular analysis using gene expression by quantitative PCR, qPCR, and NGS sequencing. Cissy, the stage is yours.
Thanks, Mina. Yeah, hello everyone. Now I'm going to talk about other downstream molecule analysis. So after the cells get enriched, isolated, and captured, researchers can also use extract pRNA for gene expression analysis. Among the common tools here, we will focus on the PCR, qPCR, and NGS sequencing. The PCR method is one of the most powerful and sensitive techniques. It's a gold standard for detecting and also the quantitating the gene expression products. It's very easy with a fast turnaround time, but the PCR needs to design and validate the primer, and it can only detect a known sequence. NGS, on the other hand, can be used to study the novel transcript and the non-coding regions. It offers a significantly better dynamic range, which is ideal for detecting the low abundance transcript. But most traditional library prep workflow is complicated and pricey, with difficulties like the data analysis. With each pro and cons, people tend to choose NGS for analyzing a large number of the genes to narrow down the research interest, and then use the qPCR as a fast, reliable, and cost-effective way to verify the NGS results. In this study, the cancer cells were first back in the blood, then captured using the Genesis system. Then the samples were isolated using Bio-Rad's pure isolation reagent to get pure and intact RNAs. The pure reagent enabled RNA extraction from a small quantity of cells. Then the 15 picogram of extracted RNA is reverse transcribed into cDNA using Bio-Rad's iScript family of ARTAS reagents. Because of the low RNA input, we pre-amplify cDNA prior to the PCR step. Remember, pre-amplification is also recommended when studying genes pressed at a low level. Then, the number of the target genes and the reference genes in cDNA samples was evaluated using qPCR assays, and the relative expression of the target genes in the both cancer and the normal samples is calculated. Again here, we use Bio-Rad's so advanced universal supermix PCR reagent and CFS Opus instrument software to conduct the real-time PCR and to do the data analysis. Bio-Rad has a wide selection of the pathway of disease-specific panels in either 96 or 384 well plate. What we use here is a SAB lung cancer panel on a 96 well plate, preloaded with the reference assays and also the control assays. The control assays include the gDNA contamination, the positive PCR, 2 of the RNA quality, and also the RT control assays. The table on the right lists all of the genes of interest detected using the lung cancer panel. 14 of them are the biomarkers from the circulating tumor cells, the CTC, which further demonstrate the efficiencies of the enrichment and the rare cell captures using our Genesis system. We had 3 reference genes to normalize RNA variation between the 2 samples. The scatter plot identified the upregulated genes in red, the downregulated genes in green, and the equivalent expressed genes in black with a 4-fold cutoff. So this is a profile of the upregulated CTC specific genes. The normal blood sample are set at 1. The blue bars show some of the clinical important genes detected in these lung cancer samples. For example, the high expression of the AGR2 is a predictive of the poor survival in lung cancer. The CAD-M1 features a small cell lung cancers. The COL11A1 is a novel biomarker in cancer research, and the CP is significantly correlated with prognosis, and the MET is a growth receptor and a predictive biomarker for the lung cancer. To summarize, the qPCR method is a gold standard. It can quickly screen those target genes that have a clinical relevance. However, it can only detect genes with known sequence. In addition, what happens if researchers are interested in other RNA biotypes besides the messenger RNA? So compared to qPCR, the revolutionized NGS sequencing technology further push the boundary of the gene study. So findings from NGS are showing the unique clinical importance, such as the detection of the novel genes and quantify the rare variants and mutation, especially the RNA-seq, which is the whole transcriptome profiling of all RNA subtypes, including the microRNAs, messenger RNAs, ribosomal RNAs, transfer RNAs, and also those non-coding RNAs become essential to understand the genomic function of both normal biology and disease-like cancer. However, working with a low abundance rare cell can be challenging. The low sensitivity, the low extraction efficiency, and the low quality of the cells all impact the quality of RNA, which can be carried to the downstream library prep workflow. This includes the poor or incomplete capture of the transcriptome, especially the small RNAs. The biased enzyme can prevent the RT from RNA into cDNA, then leads to the further loss of the transcripts. All of this will cause the unassigned transcript, the mismatched genes, or the artifacts in your data analysis. So the ideal library prep workflow should be able to reserve the rare transcript and capture the complete transcriptome correctly. Let's first take a look at the typical RNA library prep workflow. It tends to be lengthy with multiple steps involving the ribosomal RNA removal at the beginning. Then the fragment cut RNA into small fragment, reverse transcription into cDNA, adapter addition through ligation, and finally the amplification to yield a sequencing ready libraries. The ribosomal RNA depletion requires loss of peptiding. So think about how much of the sample can be lost at the beginning. Also, the workflow requires at least 3 different enzymes, the ARTase, the ligase, and the polymerase. The low sample quality can affect the performance of the enzyme. The biased enzyme produces transcription errors that produce an incomplete libraries. With all of this limitation in mind, let's see what Sequoia workflow can do for you. In Sequoia, we start the fragmentation from untreated total RNA because the sample is more stable if depleting ribosomal RNA after converting from RNA to cDNA. This is why the lost material is minimized in this post-library strategy. The RNA fragment undergoes polyidylation, creating a poly A tail on each fragment, so all sized RNA fragments can be captured without loss. The poly A tail binds to the poly DT region of the first adapter, priming a reaction that adds adapter and reverse transcribing the RNA into cDNA. Here, we leverage a proprietary retrotransposon enzyme called Zeinali to replace the traditional ARTase and ligase that couples cDNA synthesis and adapter addition in a single step. And because of the high conversion efficiency from Zeinali, our library requires fewer PCR cycles. And the last post-library ribodepletion strategy further minimize the loss of small RNAs. So the Sequoia workflow can capture more of the transcriptome, long and small RNAs. How about the data analysis solution? No problem. We have 2 bioinformatics tools available for you to analyze your NGS data, the CTCs. One is a web version that is simple to use. There's no need for complex command lines, and data is processed at the cloud. The Docker version is what you can download, install, and run on your own infrastructure. Both options will process your FASTQ file in the same underlying algorithm and will certainly give you the same result. The pipeline goes through the pre-processing for retrimming and UMI extraction, then aligns the reference genomics for both long and short RNAs in one application, then compile and normalize the gene counts to generate the report. The report shows results like the read qualities, the read trimmings, transcript coverage, RNA, and gene biotype count. Reports are in either PDF, HTML, or CSV format and can be downloaded for the downstream tertiary analysis. Sounds very interesting. So the next step we are going to do is to use the rare cell captured using the Genesis system to conduct RNA-seq libraries. The extracted RNAs will go through Sequoia complete library prep workflow and the Sequoia post-library ribodepletion workflow. The sequencing-ready libraries can be sequenced on either the Illumina sequencer or other platform like the Elements or Singular instrument. At the end of the study, we expect to show not only the concordance data of the expressed genes using the qPCR and NGS method, but also the performance of Sequoia workflow in capture both long and short RNA from the rare cell samples. So stay tuned. This is a pretty busy summary slide, but as a quick take-home message, liquid biopsy is an ideal resource for biologically monitoring. So Genesis cell isolation system enables the size-based rare cell captures, which can be used for downstream molecular applications. Bio-Res offers a suite of cutting-edge qPCR, digital PCR, and NGS instrument, reagent, and consumables, and can support your gene expression research to the next level. Thanks very much for your time. Now we are happy to take the questions.
All right, thank you, Mina and Cissy, for your informative presentation. We will now start the Q&A portion of the webinar. If you have a question you would like to ask, please do so now. Just click on the box that says Ask a Question located on the far right of your screen. Let's get started. All right, our first question here, our first set of questions, I should say, is for Cissy. Okay, what is the purpose of the post-library ribodepletion in the design of your experiment?
Great questions. So most depletion kit, deplete the ribosomal RNA prior to the library construction. The strategy carries the risk of losing the rare or limited transcript through the sample handling or RNA degradation. Our post-depletion strategy reduces the risk by removing the ribosomal RNA at the later stage. So removing ribosomal RNA after library preparation avoids the loss of small RNAs or some RNA fragment from degraded RNA samples, such as the FFPE. In addition, the benefit of pooling multiple libraries in one depletion can save the overall cost significantly.
All right, wonderful. Let's take a look at our next question here. Why is it important to capture small RNAs as well as long RNAs in a single workflow?
Another interesting question. Yeah. So both long and small RNAs are biologically important. They play a critical role in biomarker and cancer research. However, the most on-market library prep kit can only capture either long or small RNAs. Researchers have to prepare, sequence, and analyze data from multiple libraries if they want to probe the whole transcriptome. With the normal chemistry used in Sequoia complete library prep kit, customers can obtain a more holistic view of the complete transcriptome, including both long and short RNA biotypes from a single library workflow.
All right, it looks like we have one final question for you here, Cissy. What is unique about the chemistry for seq-Zyme used in the Sequoia nRNA-seq library prep kits?
Yeah, seq-Zyme is our proprietary engineered rich transposon that replaces the traditional ARTase and the ligase used in other commercial kits. Our enzyme couples cDNA synthesis and adapter addition in a single, highly efficient, continuous synthesis reaction that produce a 99% stranded libraries comprised of both long and short RNA biotypes.
All right, everybody, and it looks like our next 4 questions here are for Mina. Number one, what is the maximum volume of blood as well as the number of cells that can be run through a Celselect slide?
That's a good question. As I have shown in one of the graphs during the presentation, we have shown the sample blood volume from 4 ml to 200 microliter. Right now, the maximum blood volume that we can process through the system is 4 ml, which is going to be diluted 1 to 1 with PBS, so total like 8 ml of sample. And there were like so many researchers that they were interested in processing some animal models, like for example, mouse model, processing like very low blood volume, like for example, 200 microliter. We get the same results. However, we had some customers who were like processing 20 ml of blood sample through the system, but definitely they have used the protocol builder to be able to adopt, that high sample blood volume through the system. The second question was about the number of cells. As I mentioned, that the Celselect slide does have over 56,000 chambers. So we're not exactly sure how many cells is the maximum, as long as the chip is not getting saturated or processing, thousands of CTCs. I guess they are fine. We have shown a couple of thousand CTCs per ml using our platform, no problem, no clogging, no issues. So I hope I answered that question.
Yeah, okay, wonderful. Number two, once a blood sample is collected, how soon does it need to be used with Celselect?
Good question, and very tricky as well. So as I mentioned that normally 2 blood sample tube is being used for with Genesis. One of them is EDTA tube if you are interested in getting viable CTCs, and strict tube if you're interested in doing enumeration. So for EDTA tube, as pretty much I believe everyone knows, the cells are not fixed using EDTA tube. The sooner you process a sample, the better and more viable cells that you get. I would say up to 24 hours is good for EDTA tube, but again, the sooner the better, the more viable, the more cells. However, for a strict tube, because the cells getting fixed using that platform, they are good up to 72 hours for processing through the Genesis, and we haven't seen any inconsistency for CTC numbers.
All right, great. Number three, I am interested in doing combined ctDNA and CTC analyses from blood samples. Does the Genesis system have a workflow protocol that supports this?
That is a perfect question, and is basically aligned to one of my slides that I have shown using, working with one of our collaborators. They were like processing ctDNA using basically separating plasma, using that portion for ctDNA, and they didn't know what to do with the pretty much leftovers. We basically using Genesis, we have shown that obviously with protocol builder optimization and everything, we have shown that even after the plasma is depleted and the cells, basically the sample can go through the centrifugation and stuff, we still are able to capture CTCs, and they have shown that the enriched CTC is also expressing, for example, PD-L1 on the patient sample. So the answer is yes, you can do that, but most probably because the blood sample has been going through pretty much high centrifugation, we probably have to optimize the protocol using protocol builder.
All right, wonderful. And number four here, what fluorescent imagers or microscopes can be used for the enumeration protocol?
Yeah, so pretty much as long as you can scan the Celselect slide with any automated microscope, fluorescent microscope, you should be able to use those images toward the software, toward the analysis software. As long as they are like up to 6 channels, I would say you should be able to use any pretty much automated platform.
All right, perfect. And Mina, do you happen to have any final comments for our audience?
Thanks for the opportunity. Yes, actually, I would like to ask the audience that if they would like to know more information about the Celselect, please type info in to your chat box. And also, if you're interested in scheduling an instrument demo, please type demo. Likewise, if you like more information about our downstream application, then type dPCR or qPCR or RNA-seq in your chat box. We would be happy to help you out.
All right. Thank you again, Mina and Cissy, for your time today and for your important research. Before we go, I would like to thank the audience for joining us today and for their interesting questions. I would also like to thank our sponsor, BioRad, for sponsoring today's webinar. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speakers via the contact information you provided at the time of registration. This webcast can be viewed on demand for 2 years until May 18th, 2025. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.